them one at a time, any one of them, does not mean that you are allowed to try and get all of them and do text mining on them. And so we need to fix that, and this community needs to be active in trying to fix that. So as the sign says, please complain to NLM about that. It's a political issue. They need the support of the community to do what I think is the right thing. But in the meantime, until we win that battle, what we have is for full texts are open access journals. And that raises an important question. If we develop our tools and come to understand the way biomedical language works on the open access subset, are we biased in some way? Are we going to develop tools and techniques and descriptions of language phenomena that will not generalize to the rest of the literature? And that's, if I can get it right, good. Okay. So the tool that we use to do this, to answer that question, and a tool that we hope will help other people answer lots of other questions, is a corpus we've been working on for more than 2 years that we call Corrado Richly Annotated Full Text Corpus. The corpus is made up of nearly 100 papers that were selected because the Jackson Lab Genome Informatics project chose those papers to support at least one gene ontology or mammalian phenotype ontology Go annotation. So there's something about a gene and its function or phenotype or cellular component or biological process in that paper. And other than that, we just took a date range. We went through that corpus and have marked up every occurrence, every mention of any concept, or if you don't like the term concept, let's just say term, several open biomedical ontologies and some additional controlled vocabulary sources listed here. The cell type, chemical entities, species, sequence phenomena, the Go Big Three, and also normalized entree gene dimensions. And the entree gene part isn't quite done, but we'll get there soon. We've also marked up all of the, or many of the syntactic and linguistic, including sentence segmentation, tokenization, part of speech tagging, and building syntactic trees. This is all done by manual curators. We are in process and making good progress on co-reference annotation. So when a term in the documents refers to the same thing multiple times, when I say in 3 different places, or when I say it in some place, if that's a reference to the same thing, then we're going to link those together, an important part of text mining that hasn't been dealt with too much. We are trying, although it's going to take us a little while longer, to get coverage of the relationships among all of those other semantic phenomena that I mentioned. And we are trying to get additional funding to do more discourse analysis to identify evidence, to identify claims and hypotheses and things like that. We have marked it all up in a tool called Notator, which you can kind of vaguely see ontologies. Here can be loaded in as many as you like. You can put terms in this text, and they can be discontinued, and then associate the spans in the text with the terms in the ontology. This is on top of Protege, the frame system, and it can produce all kinds of outputs. We have a wide variety of different things we can dump it in, from XML, annotation, all kinds of things. I'm going to give you an idea of the density of these annotations. Colors code to different ontologies, and you can see there's references to concepts from ontologies in these kinds of full texts. They appear to be, I don't have evidence on this yet, but roughly speaking, they're maybe by a little bit than abstracts. It has taken us a while to get good annotator agreement, so the x-axis here is time. It's kind of arbitrary units. It's median of the annotators to resolve disputes, but roughly each integer here is 2 weeks. And you can see in the beginning we had some issues, but by the end, these are on different scales. I'll talk about this in a second. We're getting, sort of 95-ish% agreement on most of these ontologies. And the exception is Go biological process and molecular function, where we are getting sort of 80-ish. And a significant part of the reason for that is confusions between cognate terms in function and process. So the problems we're having there are not necessarily devastatingly bad, but sometimes hard to tell whether they're a reference process in the Go sense. They're double asked because they're not done with them either. And we have a fairly large total number of annotations. The Go annotations per article are captured here. You can kind of see two different distributions. So for the biological process, there are often very large number of processes mentioned in any of these articles, but by and large, the number of cell components and the number of functions mentioned tends to be a little less than 100. You can see maxima and minima here too. Whoops. I'm sorry, some of those tables covered up. Well, if you want that data, I will try and provide it. It's in the paper. We have a number of unique terms for articles. So what I showed you before is if mitochondria is mentioned 10 times, that's 10 annotations. Here, if mitochondria is mentioned 10 times, it's 1. So this is type rather than token. And distribution is similar, although a little bit more obviously bunched up, and there's probably a bigger density here for the process terms. So typically, you see about 50 process terms and about 10 locations and functions. This is now getting to that question I raised at the beginning. This is the highlight of, theoretically. And what this does is try to compare are the full text annotations that we open access sets, craft, compatible with the ones you see in traditional journals. And we can compare a bunch of different features about those. And that's, but how would we know if the differences were significant? There's no statistical way we can do this in corpora, but we can compare to an outclass, the way you would need, and the outclass we use is the Wall Street Journal set. So if you compare how different is craft from traditional journals with the basis of the Wall Street Journal, on a variety of things here, particularly with things, instructions or negatives, they're much more like each other, like the Wall Street Journal. Kullback-Leibler divergence is trying to look at, are there differences, and this is a way of trying to estimate semantic differences without having to go through and annotate all of these. And so we can look at the distribution of terms and the differences between them, and this is Kraft versus Trager, and these KL divergences suggest very strongly that these two distributions at various different sizes are indistinguishable from each other by and large. Terms distinguish best between them, and but the terms that distinguish between the craft, that's this column here, are things that make sense and are not particularly about content of the article. So it's things like PDF and record and window. But what makes them different from the Wall Street Journal and what makes traditional journals different from the Wall Street Journal, apostrophe S shows up a lot in the journal and very little in biomedical publication. Mouse shows up a lot in craft, a little bit more than it does in traditional journal documents. But by and large, these are contentful and describe contentful and say that the difference between craft and traditional. So, I think the bottom line of this is that open access articles, I'm glad I can say, are a reasonable place to develop full text text mining software. That's where we should be doing our work. And the craft corpus is, I hope, a useful tool, not just for answering questions like this, but for more generally understanding syntactic and semantic phenomena. We have plenty of evidence from other places that full texts are quite different than abstracts in a wide variety of different ways, in both structural ways, like they have far more parenthesized material, and the parenthesized material is longer, and often in semantic ways as well, and we will be able to talk about that hopefully for next year. You should look for public release of craft. We hope to have it out in January. One way to evaluate or keep track of it is to pay attention to our BioNLP SourceForge site, which is one of the places we'll put a pointer to it in. And just to, as I wind up, one of the things that we want to do is hold back some of the data that we've annotated to facilitate testing of various approaches and the like in the style of BioCreative or the CL share tasks and the like. And we're trying to design that now, so I would really appreciate it from the community as we are trying to do this design about timing, how much time do we need, about other kinds of logistical or organizational details so that we do the right thing for the community. And there, I would appreciate it if you just send email. The other, and I just want to mention this briefly, a paper that this is supposed to be a highlight of was actually not by us, but about us. It was a Nature News story from January. I sort of want to wind up by talking about what it is that full text means as we go after it. It also means that we probably need to take on more ambitious sorts of knowledge representation rather than just biology terms, which, by the way, is hard enough, or just protein-protein interactions. We need to be able to make mappings from those journal articles to a wide variety of phenomena, which fortunately are well represented in a variety of community-curated ontologies, but it does scale up not only the amount of text we go after, but the richness of the representations we need to get out of that text. Richness is not confined just to using more and more complete versions of the ontologies that we mark to, but also more complex kinds of things, like the relationships between things that can be linked together to build assertions, to be able to go after co-reference chains, because it's really important to know if we're about the same thing in multiple places, and even discourse about how people bring evidence to bear on making claims and the like. Certainly computational scaling is an obvious problem, and once you start working on large collections of full-text journals, it becomes really clear that even just fairly large clusters at this isn't the solution. We have to be smarter than that, and there's some interesting research challenges, I think, there. There has been some really good work, I think, in our community trying to make things interoperable and fit together using UEMA and now the UCompare site. That paper also came out last year, which makes it really easy to deposit your work and to then use other people's work against your own corpus, if you want, to do text mining tasks in a fair and relatively infrastructurally straightforward way. We're going to have to look about combining different sources of information about meaning, bringing additional stuff to bear on understanding these full-text journal articles. And I'm pleased to say that the purpose of doing text mining is not to produce what looks like the output of our program, to really make the results and the things that we are able to do useful to scientists who are trying to discover things and make the science better. So we have to think about what kinds of results from our text mining are really going to be of most value. And sometimes there's trade-offs between how doable something is and how valuable it would be, and we should be thinking very much about its value. I think that's it, and so with that, I hope I have time for a couple questions. Oh, lots of time. Over there?
Hi. So I have a question about automatic ontology building from this corpus. Because currently we are using an existing dictionary from Medline. Do you think it needs to be changed, or it's enough? Should some automatic ontology learning work on revising this dictionary?
I think that automatic tools can be very important in helping ontologists do their work. But I am extremely dubious of any fully automated ontology production, because what I think ontology is a community process of finding where we disagree about meaning, and then coming to talk about it and coming to resolve our disagreements about meaning. And so if it doesn't take place in a community context where people who have a stake in those terms and what they mean and how they're used are involved, kind of like a nomenclature committees, then we're not doing it right. So
yeah, I agree.
Tools are great, and there's lots of interesting tools that you could use to try and help ontologists do their work, assess the coverage, what's missing. You know, it's hard for the ontology creation community to know where they ought to put their fairly limited amounts of resources. So if we could come up with tools that would help them assess the questions that they have, or put them in directions that would be useful for them to go, I think that's great. But automated ontology construction is a non-starter for me.
Okay.
Peter.
Yeah, Larry, that's a very nice talk. It brought to mind a question, though. As you are quite ambitious in going after full-text journals, I was thinking about the experience of reading a journal article, and the text is really only one component. There are figures, there are tables, there are other structures within the publication sometimes I would only read the figure legends, for example, and because the figure's in front of me, I can parse it very easily. Have you thought of how that could be incorporated, the information content, into something that would then become useful and automated?
Thought about it. So there's a couple of answers to that. So one is some stuff about figures and tables and the like we do, right? So captions, as you point out, or legends are text, and we can apply the same techniques to that. In fact, there's an interesting history. Marty Hurst and I'm not going to remember her co-author's name, had a great project a while ago where you could search for papers using just searching through figure captions and table captions. And it was a very useful way to find material that you might not have gotten searching over abstract words and title words the way you do in PubMed. So it's clearly valuable. Second piece is it plays a role in discourse that we can probably capture with the way that we have now. So if we get to the point where, and I hope we do over the next year or so, where we're trying to capture this is evidence for that discourse relationship, then clearly this piece, a lot of it is figure 7 shows that whatever, and we can show you figure 7. The last, most ambitious piece of that, trying to do for figure understanding or table understanding what we do for understanding text in automated ways, is extremely difficult. And there have been a few attempts over the years. I think one of those things those attempts have demonstrated is that there is such a credible diversity among figures and tables that it's not one problem, it's many problems. And that we've made a little bit of progress in some of those. So there's some great work in trying to automatically pick up stuff in tables, particularly HTML tables and the like, which kind of give you some hope of being able to figure them out as opposed to and so there is some effort there to try and capture data that's in tables of papers. I think generally understanding photographs, that's a hard problem.
That's going to be hard. Another hemisphere of the brain is involved.
Yeah. Any more?
Yes.
Oh, Peter.
Yeah, I'll just put forward some more evidence why I don't think automatic ontology construction is going to work, is that there's the task that we know people have, a semantic task that we know people have a really hard time doing right. And that requires deep knowledge of the domain to get right. My belief is machines are probably going to have a much harder time doing it.
What he said. And I really do think ontology is all about a social process. It's really about coming to agreement on what terms to use and what they mean. And the point is coming to agreement. The point isn't the result of the agreement. We use the result of the agreement. I think you were next.
Hi, thanks for the good talk. I don't have a specific example, but I can think of a use case where you've got two things that look at first like they're talking about the same thing, right? But in reality, they're actually talking about different things because of the inherent assumptions about how the data was generated and so on and so forth. How are you thinking about that?
Well, in several different ways. So one is if it's the morning star and the evening star, and science has not yet discovered that they're the same thing, then there's nothing we can do about it except go back when we do discover that they're the same thing and build a owl same as or something like that between them. All right. The case where two different communities are talking about the same thing, but they use different terms for it. That's a place where I hope that text mining and ontological work can actually make some progress in science, where we can identify, point them out to people. And there's Don Swanson's work on, the fish oil and Renaud story, and from there on out, people have been trying to do that, and there's a bunch of different technical means to try it, and I think that by going to full text, we'll probably maybe do a better job of it, and certainly the work of the ontology development and annotation community helps with that too. The more difficult, I think, problem is the converse of that, by the way, which is we've been going on for a long time calling something, I don't know, cancer or diabetes, and we change our mind some years later and we decide it's really two different things. And then going back and undoing that turns out to be very hard. And, science evolves, concepts change. We change the way we phenotype things. There are going to be shifts like that. And I think it's one of the things, we can't go back and re-annotate everything. We might, with automatic tools, be able to go back and tease some of it apart. But it's going to be a hard task and problematic one that I don't see any obvious solution to.
How much time or effort would be needed for the author of an original paper to annotate it?
Well, that gets to what Peter said.
Can you aid it with, adding tools and?
No, I'm against that, and I've been against it for a long time, and I'll explain why. Knowledge representation is hard. There is a skill set involved in figuring out which ontological categories, especially when there are tens of thousands of ontological categories. We're not just talking about some small number of things, let alone relationships among them. Figuring out which ones and how they belong together and what all the borderline cases are like is really a skill. It is not something that you get easily. People need to be trained in it, they need practice at it, they need to talk to each other about it, and you can't ask authors to develop that skill set. It's too hard. There's a few things I think you can ask authors to do. I think it would be reasonable if journals would demand when an author mentions a gene that they provide a database identifier and a table, like a table of abbreviations. This is the would be really helpful, and I think authors could do that.
So, if I can...
But the Feb's letter experience was, I think, evidence that authors can't do that, even for very simple-minded stuff.
For the authors in the Feb's letter experiment, takes an hour, in average, to find the database entry that corresponds to the protein in their paper.
An hour well spent.
And
it takes me longer.
Yeah,
I mean, I can't tell what they meant.
No,
I have to. They're the ones who have the reagents.
That's fine,
okay?
but then the comment
out.
of the number of the authors was that if they are required to do this, to submit the paper to Feb's letters, they will submit somewhere else.
Well, that's right. We have to do it. All of these kinds of requirements have to be done broadly in a coalition of publishers. You can't, no one person can, or one entity can do it.
How many annotators annotate the corpus? How many annotators?
Around 20, it depends on exactly how you count. Around 20 people have been involved in the project from beginning to end. A few of them through the whole process, and some of them come and go. That counts as syntactic annotators who have had more turnover than the scientific ones.
Do you have an estimate of how many hours went into the annotation of each paper or something like that?
I do, but I wouldn't want to guess off the top of my head. I can certainly tell you, but it takes because we did it separately. So all the passes were independent. So the people who did the biological process and molecular function pass took much longer than the people who did part of speech tagging. Sure. So I could come up with statistics, but I don't have them with me.
If there are no more questions, we thank Larry again for a nice talk.